My honor to welcome Dr. Shawn Hingtgen, who is the assistant professor at UNC's Eshelman School of Pharmacy. He's also the founder of Falcon Therapeutics, and that is a company that is developing personalized neural stem cell therapies to treat glioblastoma. All right, I haven't even had coffee. He has several awards and recognitions, which really are way too numerous to name, and after talking to some of y'all, it seems like he's pretty well known. So I'm going to get off the stage so y'all can hear his presentation, and please join me in welcoming him.
All right, you hear me? Good morning, and thank you so much for the opportunity to speak to you guys today. I really appreciate it. And as Catherine said, my name's Shawn Hingtgen. So I actually have appointments mainly in the School of Pharmacy, but also because we do cancer research, so we have appointments in Lineberger. We have an appointment in the medical school to do all the neurosurgery, neuro-oncology stuff that we do. And really the whole goal of our work is to create a new way of treating cancer by harnessing the latest in stem cells. And our little catchphrase is that we can take your skin and morph it into cells that actually seek out tumors. So all we need is a little bit of your skin and a little bit of time. So before I dive into all that, I just wanted to thank you guys again, and it's really an honor to be here and kind of follow on the legacy that a lot of you guys established for us. And so without your support and what you guys did, folks like me wouldn't be able to do this. And it's really been exciting. You know, I got to know Cliff and Linda and Jean and a lot of different people here in the audience, and it's just really a pleasure to be here and kind of show you what you guys helped build and what we were able to take forwards and do with it. I give you 30 seconds about me quickly. So I'm an Iowa guy, born and raised in the Midwest, got my PhD in gene and cell therapy, so went to Boston and had the ability to go there and train for 7 years. And they had just made this discovery that stem cells could actually home into tumors. So that got me really excited, and the ability to translate that into human patients. So came here in 2012. And it's really just been an absolute wonderful experience, and couldn't imagine doing this anyplace else. And outside of the lab, we have 2 small kids, so we have a kindergartner and 3rd grader, and basically they take up my time outside. So a lot of running around to sports and different things. So really quickly, as they said, I'm supposed to tell you guys we own a company, so we spun this out in 2012, and really the main reason was because we want to get these to human patients, and having the ability to take the academic side as well as all the commercial side and use that together, it's the best way, fastest way we can get this to humans, and that's really what our goal has been. Okay, so I know it's still early in the morning, but talking a little science. So the majority of our work is around glioblastoma or brain cancer. So you guys may be familiar, I see nodding some heads. So glioblastoma, it's the most common primary brain tumor, but one of the deadliest forms of cancer. Sadly, it's never far from the news. So this was the disease that John McCain was battling a couple months ago, about a year ago, I guess, ultimately took his life. The picture we show here is Brittany Maynard. So she's 29 years old. She was diagnosed with a glioblastoma. And CNN followed her story because it was so powerful. In the end, she chose to go to Oregon and have medically assisted suicide because the options for treatment were so poor. And she basically said, I can't have my family watch me melt away from this disease. So if you think about that for a minute, how bad does a disease have to be at 29 years old to think your only option is to go have medically assisted suicide? It really speaks to the need to develop new treatments. And that's where we're trying to focus on. So just showing you a little bit about the challenges that people like Brittany face. The stats for glioblastoma, standard life expectancy after diagnosis is only about a year. And even worse, that really hasn't changed in almost 3 decades. So we really need to do better. And we think that the solution for this will come from an unlikely source, but actually will come from a stem cell. So when you think about stem cell, a lot of your mind probably goes to the place where my mind went when I started in this field, regenerative medicine. So you think about stem cells, you can do things like rebuild the spine for people like Christopher Reeve, who was Superman. You think about rebuilding aspects of the brain for Parkinson's patients like Michael J. Fox. Certainly regenerating things is a big aspect of what stem cells can do. But our work actually utilizes a little known aspect of stem cells, something that was newly discovered, and is that stem cells can actually seek out and chase tumors. So this is what my training kind of grew out of, and they had just made this discovery in Boston. And what they showed was that if you take stem cells and actually put them in the brain, they will crawl through the brain naturally and seek out a tumor that's far distance away in the other side of the brain. Not only will they find the main tumor, but they will find all the little invasive roots, all those little bits of tumor that are spread around your brain. And that's what's so hard. That's what ultimately takes the lives of patients, because clinicians can't see all those little bits that are spread around. Chemoradiation can't find those. These little cells can crawl through the brain, hunt them down, and find them. Now the challenge is, these are stem cells. They're not designed to go attack and kill a tumor, right? So they kind of find it, they'll bump up against it. So what we discovered also was that you could turn these into drug carriers. So we engineer the cells to actually pump out cytotoxic agents, things that can kill tumors. So putting those two together, when you release the cells, they'll crawl through the brain, hunt down the tumor, release the drugs really potently, so you can actually now start to kill these types of tumors that were previously untreatable. So we spent a lot of time doing things. This is hopefully is a little bit convincing you that the homing part works. So if you look at the top panels, the red up in the very top left, the red are the stem cells. That little green spot is a brain tumor. So this isn't a mouse, and we're actually imaging it a lot while the mouse is alive, and we can take pictures over time. So you can see there on the top left panel where it says day 3, the stem cells and tumors are far apart. You can watch over time in that day 7 image, all the cells, the red cells actually hunt down the tumor and start to kind of engulf it. And then by day 10, you see all the red is right where the tumor is, so they can seek it out and find it. And so some people call these kind of like tumor homing Pac-Mans, where they'll actually chomp through the brain, hunt it down, and find it. So this was some of the very first proof that these cells could actually find tumors, track it down. It's great if they can find it. We wanted to show they could kill it. So what we're showing you here on that left-hand side, the top panels, you can see the big red spot. So that's a human tumor growing in a mouse. You can see over time from day 1, it's a little tiny spot. You can barely see it. By day 13 there, you got this huge tumor that's taken up the whole brain. When we give our treatment, those are the bottom panel of mice right there. You can see the tumor barely grows at all and is almost undetectable. So that's with just a single dose. So these were some of the first studies we showed that really said, yeah, we have this thing could really work if we can get it towards human patients. So that was about that time that I moved to UNC, and the very first person I met on campus was Matt Ewan, who is the chair of neurosurgery. So hopefully I think you can see throughout the theme of my talk, I'm the basic science guy doing a lot of this in the research lab. We always want to get this to human patients, and so what's so amazing about UNC is that we have so many collaborations with the people who are treating patients in the hospital. They're so willing to give us their time and go back and forth, and so Matt is the guy treating all these brain cancer patients. And so I was here 2 days. We sat down and had a 2-hour meeting, and I said, this is what I got. And he said, let's figure out how to do this right in human patients. So out of all of that discussion, we came up with kind of 2 main topic areas that we were going to target. First of all, we said, we need to figure out how to make these cells safe and effective for a human patient. And in the end, we said, we need to make them from each person. We need to have each patient be their own source so that it's safer, better, faster, everything. The other side was supposed to be easy. He said, get these things in a format that when I'm treating a patient, I'm there in the surgery room, I can easily drop them in. And that was supposed to be simple, turned out to be more complicated, but we got to do some research and created some nice projects for us. So diving into this really quickly. So again, thinking about this, these are cells. You can think of organ transplant, cell transplant. The challenge is rejection. So we got to figure out a way to get the cells so that they can stay alive in the patient, crawl really far distances, but at the same time be really safe. And so we said, what if we can make them from each person? So talking with Matt, I said, well, could we go in and take a little bit of the brain out? He said, no, nobody's going to do that. Nobody's going to let you do it. And certainly not in a brain tumor. So, okay. So he said, go find another source. I said, okay. And I said, how much time do I have? He said, remember, these patients are really sick. I'll give you 2 weeks. You got 2 weeks to make enough. Okay, so not setting the bar very high. Well, so luckily at the same time, the latest in the world of stem cells and cell engineering and all that type of stuff had created a way to actually take your skin and morph it into anything in the body that you guys have, any cell type, and ultimately the idea was any organ, simply from a little skin punch. And so the idea was that you could start with a skin punch, you turn it into little cells that are from your skin, you use a couple genes, so you use a little gene modification, and it will actually revert your cells back to an embryonic state. So kind of you as a couple days old. And the idea is then you can take those cells and turn it forwards into anything you want. So for instance, cells from your brain. To show you the power of this, this was discovered in 2006 and won the Nobel Prize in 2012. So 6 years from discovery to Nobel Prize. So really transformative. So I talked to Matt about this, and he said, too long, too many steps. So he said, okay. So I went back to the drawing board and said, what if we cut all those steps out? So what if we could go straight from your skin to the cell that we want, and we can make it safely and in 2 weeks? And he said, that would work. So he said, let's go prove it. So pulling in some more of the cell guys, this is kind of the team we put together. So we were the very first group to theorize we could take your skin and flip it directly into cells that would seek out and kill tumors. And so that's basically what we set out to prove. So we're in the lab, we had mice, so we took mouse skin, dissolved it into cells, and then did this flipping thing where we morph it into these cells. So we call these induced neural stem cells. We've induced your skin to kind of look like these tumor-homing brain cells. And then we asked questions. So we could do imaging stuff with them to ask where do they go, how long do they stay, and we also load them with drugs to ultimately, can we kill a tumor? So that's what we set out to do. There's a picture of some of the first ones we made. They're labeled with the genes from the jellyfish, so they can light up green so we can make nice pictures. Let's see, okay. Oh, hopefully I didn't skip it. So the very first thing we asked was, can we turn your skin into the things that crawl? So normally your skin just kind of sits there. It's great, it's a nice barrier for you, but it doesn't move around. You'd have problems if it moved around. So we were trying to figure out, could our little flipping turn them into things that crawl and go find tumors? Without that, we were kind of going down the wrong path. So you can probably ultimately see where we're going. I probably wouldn't be talking to you today if it didn't work. So what we did was set up dishes and used our little imaging system to make movies of where the cells go over about 2 days. So on the left-hand side, that's an actual live image. It's a little bit hard to see at times. So the image on your right are dots where we tagged each of the cells, and you can watch them crawl. So if you can play the video. So the one on the side, it's a little bit hard to see. So the green cells are the little skin stuff, and hopefully over time you can see them crawl to the pink. But at least with the dots, you can watch the track where they all go. So that was the first time anybody had ever shown that you could turn your skin into something that goes towards a tumor. So you can see, probably the dot is a little bit easier, the path. They wander around a little bit, but all of them are going right towards that purple emblem where the tumor is. You can see they're not going the wrong direction. They're not really wandering up and down. They're all going right towards where that tumor is. And ultimately what that meant was we're conferring properties so they can sense the signals. So this was really exciting for us and really set the stage for all the work that we've been doing to move this to the clinic. So we then asked, let me see, wrong button, sorry. It's great to see it in a dish. Does it work in a mouse? So we did things where we took human tumors, put them in one side of the brain, put our cells in the other, and then asked, could they crawl through the entire brain and track it down? If you look kind of in the middle, we could see things there. So those pictures are the red tumor that's kind of in the middle of the brain. You can see all the green cells are the skin flip cells chasing down that tumor halfway through the brain. So we did a whole lot of other stuff to prove that, but ultimately between the dishes and the mice, we said, yes, we've got something that can actually chase down and kill tumors, and it actually just starts with a little bit of your skin. Again, it's great if they can find it. We need to make sure they kill it. So we armed them with some different therapies. This one we call Trail. It's easy to say and works really well. So the cells are now pumping out this drug. They're actually making the drug and releasing it all at the same time. So what you can see there on the bottom left, those are pink tumor cells with our control, our little green stem cells. When we turn the drug on, all the pink goes away. The little bar graph there, you can see the lines are much lower in the treatment group. So we kill about 90, 95%, maybe even a little bit more in a dish. Again, dish is okay. We think the mouse is better. So the top left panel, you can see the mice. That we were doing imaging to track tumor growth. So really, if you look at that black line, it goes up really steeply. The tumors are growing like crazy. So we get a couple hundred-fold growth of that tumor. But when we treat it, which is that red line, it stays completely flat. So the tumor did not grow at all when we dosed our cells. To give a little visual, the things on the top right are a little pink. So you can see that's an outline of the mouse brain. The pink tumor is almost half the brain. We can shrink it down to just that tiny little spot with a single dose. And then the graph on the bottom is how long the mice survive. So basically what that means is we almost tripled the survival with a single dose. So we were able to publish that paper, and it was the very first time that anybody had ever used this. So we're able to, with the UNC team, put some patents down and really start investigating how do we move this to the clinic and get it out of the lab and into the hands of clinicians like Matt who could start to use this to treat patients. So the first step, Matt said, of course, was, mice are great, do this with human skin because we need to move forward. So we did a lot of the same things. I won't bother you guys with a lot of the data. I'm happy to talk about all the stuff if you want to see it. But so we did human patient skin. So we're taking biopsies from human skin. The other thing he said is, remember my timeline, get these things to me fast. So we were able to show that the flipping process worked in about 2 days. So we're compressing down to that real short time frame because a lot of the other people in the field for regenerative medicine, Parkinson's things, they're not working in such a tight window. So we had to create a bunch of new technology to go really fast because these cancer patients are really sick. So we were able to do the flipping really quickly. I won't bore, it's a lot of genetics. If you guys want to know about genetics, I'm happy to tell you about it. Long story short, if you look at the bottom left panel, there's a red dot down there. That's your skin. On the right-hand side, there's some blue dots. Those are cells in the brain. And you can see there's a little curvy line. So our cells are the green ones. They're in between all of it. So ultimately, it said, we've created an entirely new type of cell that's not skin. It's not your brain. It's this unique carrier that has these special properties that can seek out tumors and deliver drugs. And so we're kind of working through characterizing what all that means. How do we make them work the best? And ultimately, how are they safe and efficacious in humans? So building on that a little bit, we had to show that they home. And so these are two little blobs of cells kind of in a three-dimensional matrix. So the green is a tumor. The red is a bunch of our little cells made from human skin. And hopefully you can see over time as you go left to right, that little blob moves into the center of the tumor. So we could actually show in kind of models that made better pictures, these cells will actually crawl, penetrate, and chew into the core of a tumor. So just like the mouse cells, these things will home, they'll penetrate through tumors. And so again, it works not only in mouse skin, now human skin. When we release the drug, we can see basically the bottom graph there. The lines are really far apart, so we're getting really big extensions in survival, just like we saw with the other cells. So the treatment overall is working. We about double or triple the survival again when we do these in mice. We did another little advancement with Matt, which we can do much better now, but he said, remember, I'm a neurosurgeon, so surgery's key. So we scooped out the tumor, did surgery like he does, and dropped our cells in the cavity to start getting a little bit further even towards that clinical trial. And so you can see again here, out of all that stuff, in the bottom right, the lines separate again. So even kind of moving towards what this would look like in a human patient, the treatment is still holding together and still working. So this was about 5-7 years worth of work, and ultimately in the end, it's probably easier to show you the picture. We're envisioning an entirely new type of treatment and a new clinical trial. We're putting this all together right now where a brain cancer patient would come in. We would actually start with a skin biopsy, take the biopsy, dissolve it into cells, do our little engineering, flipping, and make the drug, and then we'd go back into the same patient for personalized cell therapy. And the cells could then hunt down the tumor and kill it. And so we really think this is a way we can start getting some traction against these diseases like glioblastoma. We've done this for pediatric diseases. We've done this for metastatic diseases, things like melanoma, breast cancer that goes to your brain. And so we're really trying to just see how broad the platform is as well. Again, what's so wonderful, having everything close by, we're getting patient biopsy samples. So people, GBM patients that are undergoing surgery at UNC Hospital, we're getting parts of their tumor and parts of their skin, which took a long time to explain why we need skin from the brain cancer patients, but we got them both now. So we're doing testing on taking a patient's skin and tumor and putting it back together in the same dish so we can actually show this works. So continually working to try to drive this towards the clinic. So that was kind of the first part. I just wanted to tell you a little bit more about kind of the second part quickly, and then happy to take questions or anything you guys want to know about. So again, thinking with Matt, he said, okay, so you solved my first problem. You got me cool cells that work really well. You got to help me make sure I can get these into a human patient. You know, the OR is busy. We got lots of moving parts. We need to make sure that this is a simple process as well as safe for the human patient. And he said, so remember the standard of care, so the top little panels there are showing if a patient comes in with a tumor, you have a big red tumor, he said the first thing we're going to do is take the tumor out. So you're going to have this cavity there, and then your therapy will probably go in that cavity because the idea is you got to stop the tumor from coming back. So do some testing, figure out how we can make a cavity, how we can get the cells in there and get it to work. I said, well, here's the problem. The bottom shows what we can do in the lab. We basically make a big tumor and stick things in there. So he said, well, go make me a surgery model in mice and then show the stuff works in there. So I said, okay. So we got Matt in the lab and we did some different things. The first thing we did was take mice. We put human tumors, we labeled them green. So this is looking down on the top of the brain of a mouse. You can see the green tumor actually showing through their skull. So we said, okay, we'll take the bone off, and then we'll actually do brain surgery in these mice. So again, we got these tumors, and then we went in, and we, with Matt's help, actually were able to resect the tumor just very similarly to the way they do this in human patients. So can you play that movie really quick? So you can see basically there's a little ball there, and we're actually using a vacuum. So the little black thing you see coming in is the vacuum. It's about the size of a pen tip, maybe a little bit smaller. So all of this is done through a microscope with a mice under it and all that type of stuff too. In the end, that tumor will come out, and it leaves little bits of tumor in the brain, which is exactly the same thing that happens in human patients. They can't quite see all of it, so they get a lot of it, but not all of it. Do you mind playing that one really quick? So this is where the tumor actually comes out, and then we're left with just kind of little bits behind. So just sidestepping quickly, to show you that the surgery actually worked, the left are pictures, the top is our little light imaging, the bottom are MRI. So you can hopefully see lots of tumor on the left-hand side panels, and then it goes down. The graphs show you we got about 95% of the tumor out. My fear was that if you take 95% of the tumor out, it's never gonna come back, so it would the rest would kind of fade away. Quickly, turns out it comes back extremely fast. The entire tumor regrew in about 2 weeks, 13 days. In the very, very bottom right panel, hope you can see, that's the side section. It's an MRI through, and kind of on the top there, right under the word day 13, there's a big white mass. That's the tumor completely regrown. You can see it's actually coming out of the brain. So just kind of shows you how aggressive these tumors are. And the surgery can even kind of speed things up in a little way, which was a new discovery. And we're working with Matt to understand kind of the impacts of surgery on this disease. But long story short, we really needed it, so we had this cavity. So we had a cup so we could drop our cells in it. So we went ahead and did that. In talking with Matt and looking at other clinical trials, most people were just taking a needle. So you make your fancy cells, put them in a little needle, they'll inject them into the brain. Made sense to us, so we tried it. Turned out that, so the little graph there, I'll walk you through. So the black line showed the way that our cells had always worked, how many cells stayed there, how long they stayed. So you can see the line stays pretty flat. So the cells were happy in the brain. When there no surgery, everything was kosher. So I thought, well, the same thing will happen. Turns out, when we did surgery, just by doing surgery, we injected the cells the same way. The red line shows how long they survive. So hopefully you can appreciate that line drops pretty quickly. So basically, we went through all that hassle, all that work to make the big fancy skin flip cells, and putting them in the context of a patient, the surgery wiped them all out. So we're turning back to the lab to figure out why. In the meantime, it basically meant that we lost so many of our cells that the tumor came back at the same rate with or without treatment. So we ran into a serious clinical hurdle here because we had no way to get them into a patient in a safe and practical setting, so they worked. So we called our folks down the street at NC State because we turned to kind of a biomedical solution and just said, can we fix this? While we figure out why it happens, can we fix it so we can keep moving to patients? So NC State has done an amazing job of making basically what we call scaffolds. So they look like notebook paper, but they're actually made out of material that would be implanted inside of us. So bone, pins, sutures, bone screw, that type of stuff, and they can actually weave it. They melt it and they can weave it into material that looks just like notebook paper. And so the idea was, could we take these degradable scaffolds and basically make a little bandage? So we coat our cells on it, and then we hand this to Matt, and Matt would kind of just drop that in the cavity. So you would like wallpaper or line the cavity with it. So it kind of holds them there, but lets the cells still do what they need to do. So nobody had ever done this for brain cancer, but that's what we set out to do. So the idea again, so we'll take our little green, which are our fancy cells, put them on the blue little matrices, and they go into the cavity and literally wallpaper kind of line the cavity with it. And hopefully that'll keep them there and let them go kill the rest of the tumor. So this is what it looks like. So we can make them different sizes, maybe the size of a dime or a quarter. We can cut them to different things. And hopefully you can kind of see in the bottom right, it's kind of woven together. So they literally have a needle and these little electric field that draws it, and they kind of weave it all back and forth and build it up, and eventually it looks very much like notebook paper. So we got our cells stuck on there. And we asked the big question, does it help? Does all that work actually keep more of your cells there? And so the pictures on the left, basically you can see that green, that's the scaffold. The cells are labeled green, so it's sitting on top of that material. This is now sitting in the brain cavity, so they're inside. The bottom right is the graph of how long the cells stay. So If you look at the black bars, that's the old way with the needle. So everything, the black bar disappeared within 2 days. It was 7 days, so really quickly. But you could see the red bars now, actually with the scaffold, we got more of our dose in and the cells actually stayed. So if you look at the very right, by whatever that is, a month, we still have half of our cells there. So this kind of engineering partnership with NC State really allowed us to overcome that loss, which was really unexpected, and kept us going on the path towards the clinic. So we were able to keep the cells there. The problem is, could you play that video quick? So they really liked the matrix and they didn't want to come off. So you can see that a little bit. They don't move, right? So, so you solve one problem and another one comes up. So we said, okay, that's great. They stay there. So we had to go back and tweak and change the material a little bit so that they could go find the tumor, because that's really what was the important part. So we tweaked the material. I apologize. The bright thing in the middle is a bubble. There's always a bubble in your imaging field when you want your pretty picture. So these are our cells on the top in green, and now these are human brain tumors in red on the bottom. So if you play this one, after we were able to do some tweaks, you can see all the cells crawl off and then go find the tumor. And so that was really exciting for us. We were able to kind of take that leap. And if that one plays again, you mind playing that one once more? If you look at the red, you can watch, these have the drug turned on, so when they crawl down and touch the red, all the red goes away. So it kind of shows you that tumor homing Pac-Man combo. So this was really exciting for us. Putting it all together, we did the final mouse study. Basically, the bottom left, the graph show you the light gray bars are much lower than the black bars, so we're again able to stop the tumor. And this time it's the tumor from coming back because we've actually resected it, so it slows it down. We're able to double survival on this. We were doing much better now, so this was about 1.5 years ago, but it was our first kind of proof. We're now on version 10 with this, so we're making something that's safer and that works even better. And so putting that all together, what we're really trying to do is create this new trial again where skin punch, do the flipping, loading, and now we'll put it on a little matrix that Matt can then line into the surgical cavity. And so as we move towards the clinic, we've also expanded our partnership with NC State. We're doing these in large animals and different things to really try to make a full clinical dose and do it in a format that really looks much more like a human patient. So those are ongoing right now and really exciting for us because it looks really promising so far. So just in the last couple minutes, hopefully I didn't go over time, the things we're really exploring now, which is a new direction for us, are cancers outside of the brain. So in looking at the technology, the cells have these sensors that are detecting signals from the tumor. It turns out that a lot of different tumors give off these signals. So we thought, could we actually use these very same cells to hunt down other types of tumors? So we've been looking at breast cancer, lung cancer, different types of indications. And the power to this would be all we would have to do is inject the cells in your bloodstream. So just a little injection, they would hunt it down and find it. And so some of the first work we've been doing is on breast cancer. And so the very first thing we had to ask, again, just like the brain, was do these things find tumors? And now for us, it meant can they home through your bloodstream and populate that tumor that's really far away? So we put human breast tumors down in the fat pad. We inject the cells, and in this case for mice, it goes through their tail because it's the biggest vessel. So instead of it would be your hand or your arm for a human, it's the tail for a mouse. And then we imaged where do they go. So hopefully you can see in that bottom left graph there with the mouse where he's laying, they have a fat pad tumor there, that bright spot is the tumor, and you can see that is the accumulation of the cells at the tumor. The red and green pictures are basically showing a big red tumor. The tumor is bright red, and you can see all the little green cells that have accumulated around it. So it was able to find that tumor and localize at it, even though you gave the drug here in your hand, the tumor's in a completely different location. So it suggests that now we can not only treat brain, but other types of cancers. We did some new therapy. This stuff's really ongoing. Our guys are working on it right now. But you can see in the top panel there, that's human breast cancer. The tumor's growing. And in the bottom one, we can drop it below detection. So I don't think we're wiping it out. I think we're making it so small that the imaging can't see it. So we're really working hard to push this for different indications. Again, lung and breast are the two that we have grants to push forwards to. So we're really working hard on this as well. And just kind of in closing, I just wanted to say this is truly a team effort. These are pictures of the Marsico, the Research Institute, NC State. The bottom right is the hospital. And again, for every one of these projects, it's been so amazing because we've been able to go back and forth with clinicians. So on the brain cancer, Matt is the chair of neurosurgery. Simon is the head of all oncology. Those guys weigh in all of this and just make everything better. Carrie Anders, who just left for Duke, but she's set us up with an amazing team on the breast cancer side. Lisa Carey also, they're feeding in, so the people who are actually treating the breast cancer patients are helping us develop this tech, so it just makes it so much better. And the partnerships with NC State for engineering and now large animal trials, it's been wonderful. And then on the bottom left, I don't know if you guys are aware, but Lineberger has just spent a significant amount of money to build a cell therapy manufacturing center so they can actually make cell therapies like this. So this is kind of these crazy new next generation things. UNC has, I think there's only 10 maybe in the country, and I've heard that ours is the largest, and I believe it in the entire Southeast. So maybe Boston and California maybe have bigger ones, but so we can actually take our cells and manufacture these right now because UNC has the capacity to do that. There's a number of other trials that are actually ongoing already in humans where they're doing blood draws, CAR T, these kind of crazy things that are really showing promise. UNC is at the front of that because of those types of facilities. So it's a really exciting place and just gets us to the clinic faster. And in the end, a lot of people to thank. I can run through those. We have a lot of collaborations, and that's our group. So really, it's these guys. Part of too is just having wonderful students in such a great atmosphere that people want to come work. So we can get great people in to really push the science and really move this forwards. So. And again, we're over in Marsico, so we're always happy to give tours to show you guys the research space. So if anybody's interested in seeing the research facilities, they're really incredible. So happy to host you guys, or always happy to talk about science. So I think that's all I had. Thank you. Thank you.
Thank you, Shawn. For the folks who weren't here at the earlier session, to help manage and answer as many questions, if y'all have questions, if you could write them on, we have paper on the table, write them and then just hold them up, and myself or Jason will come and pick them up. And we have our first one. Okay. Are there any similar studies being conducted for pancreatic cancer?
Yeah, it's an interesting, pancreatic, it's such a terrible disease. It's on people's radar. So clinically, there have not been, but preclinically, there are people looking at it. So one of the challenges with pancreas is the fibrosis that arises around the tumor. And so we've thought about it a lot. We're actually working with some of the clinicians here to get the right model so that we can model the fibrosis and then see if the cells crawl through it. So clinically, I'm guessing in the next 2 years, there will be a trial that will be similar for this. For the stem cell stuff. Now, the other big thing is the CAR T, where they pull your blood, and then the challenge is tumors hide themselves, so your immune system can't see the tumor. So there's this new technology where they actually pull your blood out and take your immune cells and then engineer it so it can see it and then fuse it back in. So those are already in the clinic, and they're looking at a lot of different indications. So my guess is there probably is those are called CAR T, and my guess is there's a CAR T therapy probably already in clinical testing for diseases like pancreatic. Yeah. No, thanks.
When do you anticipate these therapies rolling out and to start saving human lives? And then how can we as alums assist you and your UNC colleagues with this effort? And do you anticipate winning a Nobel Prize?
I'll start backwards. Sure, it'd be great to win the Nobel Prize. Honestly, in the end, I talked a lot about this with my family. I mean, if we can get something to patients that would save even one life, I think I would feel successful as a successful career with that. So all the other stuff, I mean, it would be great, obviously, but our really goal is to get these to patients and really help people suffering from these diseases. And to me, that's an incredibly rewarding career. So let's see, timing and something else in there too. So the academic stuff is great. Ultimately, these take money. I mean, they're really expensive to develop. So that's where the company has helped raise money and move this forwards commercially. So raising money and moving towards the clinic. As of right now, today, our first engagement with the FDA will be in about 5 weeks. So we spent a lot of time working to that. So we're putting the packet. I was working this morning writing all the sections to kind of help with that. So as of right now, we'll do that. And then So you engage the FDA kind of early. They kind of give you a, yeah, your general plan looks good. Then you got to go execute the plan. And so it'll probably take us a year, maybe a year and a half if we need a little bit of buffer there. And so as of right now, we're about a year and a half, but getting a more realistic time frame because we're getting closer at every step. And what you can help as a woman, in the end, it's just support. So, spreading the word about this, and we're always happy to engage. I think everybody has a different path forward. So, come talk to us. We're happy to see where interests lie. And in the end, it's really a team effort across so many different fields. I mean, you guys saw the research side, but there's so much that goes on. Patient advocacy, awareness, so many other resources to do this together. And I think our technology is great. There's a lot of other people that have great stuff too. So I would say come talk to us and see how we could work together to make it better.
Someone then had a question if you see any application of this for prostate cancer?
Yeah, we've looked at prostate too. So in the end, we've tested about maybe a dozen different types of tumors. The cell's home to all of those, and so now really it's more of a bandwidth issue of which ones do we move through. So I'm sure there will be some where the cells don't home to. We haven't found those yet. And some of this comes to, so being in the School of Pharmacy, we're extremely lucky. Fred Eshelman gave the $100 million gift a few years ago. And so we have the opportunity to apply for those grants. And so that's been a really good mechanism for us to test these really crazy new indications. So we have a grant for lung cancer, we have a grant for breast cancer, and then we just put in a new one, which we're supposed to hear next week on some new kind of crazy tweaks and twists. So that really is kind of helping us prioritize our indications. And some of it comes from the clinicians. They're busy guys, and different people have different availability. So we're really lucky to have a lung cancer clinician, and then the breast cancer team is amazing too. So in the end, yeah, I think it has a really broad application. We're just need more time and more resources to work through all the indications.
No pressure. Someone then asked, is the stem cell research at UGA similar to what UNC is doing, or is it different?
I know a couple of the people there, so similar and different. Ours is a little bit of a unique thing because what we're doing is really applying stem cells for drug delivery, so it's a little bit different. A lot of the traditional stem cell research and even the biomedical research is more on regenerative medicine, so building organs, taking those types of scaffolds and making artificial skin, patches for the heart. There's a lot that are making artificial knees and cartilage and bone replacements. There's some really cool stuff. I know UGA, Virginia Tech, all these guys, they're tremendous biomedical engineering facilities, and they're working on more indications like that for all sorts of different applications. So there's a group at Harvard and a group at Northwestern that are kind of doing sort of the same stuff with the stem cell homing, and we work closely with a lot of those guys, but we're the only three right now that I know of that are doing kind of that application. So I would say similar but different, yeah.
This is a three-part question. How long do the stem cells live?
Yep.
How often would you get injections, and cost?
Yeah, so it's interesting. So I don't know how much regulatory you guys want to hear about in the morning. So the challenge is, in the end, this is a personalized product, right? So for me, it would be my skin back into me where I think the cells would last a long time because our immune system wouldn't recognize it. Now the challenge with answering that question, and really the questions for the FDA, how do you test that before you get to a human patient where you take their own skin and put it back in? I showed you my study, so I can give you an answer in a mouse. They'll last about 10 days to 2 weeks if we put a human cell in a mouse, even in a mouse that has only half an immune system. Ultimately, the mouse's immune system says, oh, these are human cells, they're not mine, and they get rejected. So there are honestly no good models to predict that. That's where some of the work with the canine trial came in. So each canine is getting a skin punch, and then the cells are going back into them to try to predict how long it will work. In the end, other people will argue, well, that's a dog, that's not a human, so there may be different. So really the challenge is, I can tell you in a mouse about 2 weeks. In a human, we don't really know fully until we actually test it in a human patient. And that's some of the kind of craziness that's going on with a lot of these new therapies. There are just you don't know until you actually treat a human. And so we're working with the FDA right now to put the safety plan together. And so we'll have a better answer once we actually get into human patients.
Is there any collateral damage to the other cells other than the tumor, and is there future inoculation opportunities?
Yeah, so safety is always paramount. I mean, that's really what the FDA cares. I mean, they care if it works, but ultimately it has to be safe. So we have not seen collateral damage in the testing we've done. We have to do a really big definitive safety study for the FDA. But part of the reason we really like this technology and, talking with the clinicians, they said, first and foremost, it has to be safe. We will not put this in anybody if we don't feel it's safe. So we thought about safety from the beginning. So the cells, the homing gives them localization at the tumor, and the drug is targeted specifically to the tumor. So it helps us reduce any collateral damage so the drug isn't floating around. Like one of the challenges with chemo is it kind of goes everywhere and can be taken up in a lot of other tissues. Our cells, go straight to the tumor, and the drug is localized mostly to the tumor. So we're trying to minimize that. So I would say, as of right now, we've not seen collateral damage, but we need to do a little bit more testing before I would say there's zero. There's never really zero. There's always very, very, hopefully it's very minimal, so we have no effect in humans. Inoculation. Yeah, that's interesting. So we're exploring that right now, more like a vaccine where we could prevent some of these things. I think there's probably some other technologies that would be better at a true vaccine. We've thought about things where if you had a way to predict a patient maybe at a very early stage of tumors or somewhere you could kind of have an early surveillance, we could send them in early. It probably wouldn't work. Ours probably wouldn't work for a true inoculation where you would maybe grind up the tumor and create like a vaccine. But I can tell you there are certainly really good technologies in the labs around us that are creating vaccines like that.
What do you think? Why do you think the treatment doesn't totally eradicate the cancer? And are any thoughts on using this for Alzheimer's?
Alzheimer's, sure. I love the first question. So we actually got our first big grant from the government, from NIH on that. Well, why does that spot not go away? We think there's 3 reasons. So one of it gets back to the persistence. So the cells are getting cleared fairly quickly because they're human in a mouse. So maybe they disappear before they can fully eradicate the tumor. Some of it may be that that goes together with tumors that are really far away. The cells just haven't had time to get all the way through the brain to kill it all. So, and then the third one may be resistance. Tumors are really heterogeneous. They're a whole mix of cells. So there may be some cells in there that are just not sensitive to our single agent drug. So with that grant, we're actually working through that. So we're working on things like redosing so that we can put the cells back in and kind of make them stay longer. We're swapping drugs and doing all sorts of different things. So one of the guys in our lab just finished a study where we actually take rats, we take their brain out and slice it like a loaf of bread really quickly, and you can keep the slices alive for a few weeks to a month. And so we can actually grow tumors and do all sorts of stuff kind of as a hybrid, so a model that's as good as a mouse, but much easier and much faster. And that data looks like it is a combination of both. The cells are clearing quickly, so if we put them back, they can keep working better. And if we swap drugs, we can get better tumor kills. So there'll be more to come on that for sure. And Alzheimer's, that's more of a regenerative application, but potentially. We've been focused really closely on cancer, but there's definitely research on using stem cells and this type of stuff for Alzheimer's. And actually, I would say the imaging center here does a lot of work on Alzheimer's and degenerative diseases, so there's lots of opportunities to explore that. We haven't had the bandwidth to quite go that direction yet.
And apologies, I can't read the last word of this question. Can any of these therapies be used for small cell?
Lung, yeah.
Oh, thank you.
We're working on it, so we're pushing. So we have models where we can put in tumors, we inject, and we can see the cells go to the lung tumors and then start to treat it. So we started with non-small cell. Just some of it was the lines we had available to test, and so we're working really closely with Chad Paco, who's a lung cancer clinician here too. So preliminarily, I would say yes, the data looks good. They can get there and they can kill tumors. So fingers crossed that pretty soon we'll have something ready for it too. But that's one of the main indications just for many reasons. So, and having the clinicians on board will get us to the clinic faster and in the right format. So, yeah.
Stem cell therapy for knee surgery, especially ligament regeneration?
Yeah. Definitely. We won't be the ones that get there first, but I can tell you quickly. So NC State is really working heavily on that. They have an amazing team at the biomedical engineering division there. We have a joint project where they're actually building a robot that can fabricate these scaffolds in 3 dimensions. So it's a robotic arm that will make these scaffolds. And The thing that they've done first is to make cartilage and bone. So 2 of the projects that are on it are for knee regeneration. And so one of the things, like the meniscuses are curved, so they can have this robot make this kind of thing that looks exactly like a meniscus for knee replacements in different joints. And then they could put stem cells on it that will actually remake the cartilage and the bone. So we're looking at it to use the robot to make things for the brain. But those guys, it's amazing stuff. And they're using all the resources they have there with horses and different things to really get it on a human scale and move it forward. So NC State is the place for that. It's a great place.
How did they get you to come to Carolina?
That's an easy. It's a great place to be. You know, we looked around, so we were in Boston and trying to find the right place, and it was really a combination of what was best for the family and best for the career. And so really looking at what we do, we're kind of a weird hybrid. So I spent my entire life in the School of Medicine, but really what we do is drug delivery. We're making delivery systems. They just happen to be stem cells. So if you're aware, so Carolina has one of the best drug delivery schools in the nation. So our division is about 12 different drug delivery people only focused on drug delivery. So some people use viruses. There's a couple of us that use cells. People are using these nanoparticles where they're literally, you know, like 1/100th the size of a human cell. And so we're one of the only schools that has that kind of concentration of people. So it was a really good fit for our research. The other was the clinicians right there. I mean, we wanted to be translational, so having access to the hospital across the street from us and people really willing to work with us, and the imaging. I mean, it was like a match made in heaven for us, and I was just lucky they were willing to take me on. So it fit really well, not to mention, which all you guys know, I mean, the weather's so much better here than Boston. Cost of living's a little better, so we've just really enjoyed it from the family side too, so yeah.
In the future, would it be practical to think of a preemptive strike to implant the green cells even before diagnosis of cancer?
Yeah, it's interesting, and that's we're interested in, some of it just hasn't been able to come to fruition yet, of looking at biomarkers, things to predict how early could you detect it, and then could you send these things in very early. So absolutely, it's an interesting field because you need people that can detect the tumor. So you're combining genetics with little engineering devices that can detect really, really few cells and just get it all moving. So that would be the ultimate goal too, is if you could even preemptively, before people even have symptoms, go in and wipe it out before the tumor even arises. So I think we're there. We need a little more work on detecting what those indicators are so that we don't send them into people who ultimately don't have cancer. So we just need to get that right before we get there. Yeah.
I might need an interpretation from the person who had 2 questions. The first one was, is the brain surgery necessary? And then, is it any toxic implications? I'm assuming it's not tax implications for, I don't even know, something with chronic. Here. You seem to have a very specific question, so I'll let you.
Sure.
Yeah, it's any treatment implications for syringomyelia with Chiari malformation.
Oh, I can't believe I didn't get that.
I'm not sure about the last one. I'll get Matt up here. He can tell you about that one. So is the surgery, not every patient gets surgery. There are some people who have tumors in places that you just can't access, so speech. So it really comes down to a quality of life. But about 60% of the patients get surgery because taking the tumor out, so the reason they're having symptoms, stroke, seizure, that type of, is because the tumor's pushing against their brain. So relieving that pressure is really beneficial and buys time for them. So they tend to lean towards surgery if they can. So there's certainly patients that can't have surgery because the tumor is not accessible or in an inaccessible region. So for those types of things, we're creating another way where we can actually infuse the cells and it'll find it without a surgical system, just to kind of cover that too. So the specifics on issues from the surgery, I'd defer to the clinician, so I'm happy to have Matt connect you with Matt and shoot him an email. He would know off the top of his head. In mice, no, but that's all I can tell you, sorry.
Someone then asked, was Jimmy Carter's brain tumor a is it glioblastoma?
Yeah, I should know that.
And if so, what type of therapy was used to successfully?
I don't think it was, because I don't think he had chemoradiation. I should know that. I don't remember off the top of my head. Okay. He's done such a good job he's had such a long survival that statistics would say probably not, but I should know. We've talked about this and I don't remember off the top of my head. So I think he had a metastatic, and I can't remember if it was melanoma or something that went. And so they would treat him melanoma. Yeah, so they would treat more with a melanoma cocktail, even though it was in the brain for that. So, yeah.
I might paraphrase this one. Someone had asked about the Physicians Committee for Responsible Medicine.
Okay.
And it says it's using research that doesn't involve animals, which
sometimes translates to human beings, I think is what it says. Where does UNC stand on this?
On using animals that can
Was
that can support research into, or using human tissue in animals or animals to support research in humans. I think that's fair. Sorry, I was supposed to repeat the question, which I failed miserably on. So I think that was the question, is kind of back and forth between animals to humans and humans to animals. So hopefully, if I don't answer this, grab me. I'm happy to give a better answer to whoever asked that. So the animal to human data, so the animal data is all supported for humans. So I mean, if you're going to go to the FDA is going to require animal testing to prove that it's safe and to prove that you've got the right dose and all that type of stuff. So that's kind of a more traditional model where you're doing dosing up and down and specific time points and all that. So that's the data that goes in to support your first in human trials, and the FDA has to give you the guarantee. You have to mostly prove that it's safe before they will ever let you put that in a human. And so I think that's probably more what you guys think of as a traditional pathway to getting a drug approved for first in human testing. From where does UNC stand on more like where we're getting biopsies and stuff? So we have to write protocols. So there's an institutional review board that reviews all those protocols and reviews it very heavily. It's a little bit lighter because we're not actually putting a drug in a human here at UNC. We're asking for tissue that's being taken out as part of a normal surgery. So they do review it. They need to make sure it's safe, and we really have to prove that there's no extra harm or injury to the human patient. So since we're doing this as part of a normal brain tumor surgery anyways, we're basically collecting tissue that would go to waste anyways. But there's all sorts of rules and regulations around data tracking, what we're allowed to know about the patients. We know very little. So there's patient safety. There's all sorts of regulations around that material because it ultimately is the property of the patient. So it's a really rigorous process that's controlled very closely.
Do you
have one more? Sure. Yeah, which I think we spoke a little bit about already. Just said they have a relative who has brain cancer already, so how close are we to the clinic? So again, never fast enough and never close enough, but as right now on the current pace, we're a little over a year away probably. And just what was surprising to me was, we did a good job in the lab. We've got a great team really making these cells on a clinical scale and doing it. There were so many moving parts to it. Getting it all sorted, it took longer than we expected, and getting all the safety data just took longer than we expected. So I can promise you we're working as fast as we can. It's never fast enough for patients who are diagnosed with this. So we'll keep pushing. Yeah.
My understanding of brain cancer is it is typically terminal in a short period.
Yeah.
The ethics of this would seem to suggest that you be given more of a chance to try out your therapy since the patient's going to die anyway.
Yep.
And if your data helps the next one, that's good.
Yep.
But if it cures that person, even better.
Yeah. Yeah, so the point was with diseases like GBM, where the mortality rate is so high and the diseases are so aggressive, doesn't that argue that you should have more flexibility, I think, in getting these types of treatments to patients because they need it and it will be informative for the next set of patients, and that's absolutely true. So again, we're starting to have those discussions with the FDA, and our consultants have told us that the FDA does take into account those types of things. They will give you've got to show it's safe, but they'll give you a little bit easier metric lots of times because they understand the urgency and the need for these types of things for these types of lethal cancers. So, I mean, it's awful for patients, but on a drug delivery side, it is a little bit, it does speed the process up a little bit just because there is such a need for it. So you're absolutely right, and they do take into account that. But in the end, the bar is still, I mean, you've got to show it's safe, and that takes a bit of time. There's kind of a set 3 months, 6 months, 9 months, depending on how, approval process to prove the FDA it's safe, and then they'll let you go do it. But you do have a little bit more flexibility once you're there. It's a great point. Yeah.
I'm going to, before we thank Shawn for his amazing presentation, one housekeeping note, the luncheon does start at 11:30, so if we could calmly, but swiftly, not swiftly, but, just take your time heading out. It is a large crowd. I mean, we had over 800 folks RSVP. So to just make sure everyone has time to use facilities, get to where they need to go in a timely manner. Just when we're done here, head over to the Hill Alumni Center. Douglas, Jason, and myself, when I'm able to, will help direct you if you're not sure where to go. But Shawn, thank you. That was
no.
just really amazing.
No, and